hours.
So, today we're going to teach you something about DNA replication, transcription, and
Translation.
A scientist, Rosalind Franklin, used the X-ray diffraction to define the double helix structure of DNA. He discovered that there are two strands of phosphate sugar backbone with nitrogenous base twisted together. Every single unit of DNA is nucleotide, which consists of one deoxyribose, a kind of sugar, bonded with a phosphate group and a nitrogenous base, opposite to the phosphate group. The model of DNA was first constructed by Watson and Crick in 1953. Okay, let's look at DNA replication process. When you karyotic cell doing replication, the process of DNA replication occurs in S phase of the interphase, and this process occurs in nucleus. In 1958, there are two scientists named Michelson and Stoll conducted an experiment with N14 and A15, which is highly reactive isotope of nitrogen, to identify the replication pattern of DNA. Well, they first put E. coli into N15 medium for a while, then transfer the bacteria into N14 medium and centrifuge. They find what appears in the centrifuge second generation E. coli is two strands of DNA, which one of them is purely consists the N14 and the other is a mixture, which proves the semi-conservative replication of DNA. Now, the DNA replication. The first step is to unwind the double helix structure of DNA. Helicase is a kind of enzyme that uses ATP to move and unzip the two strands of DNA. Single-strand binding protein attached to each strand keeps them open. DNA polymerase III catalyzes the formation of new strands using DNTPs. DNTPs stand for deoxyribonucleotide triphosphate. By pairing the DNTPs to their complementary base pair from 5' to 3', the leading strand was formed. On the opposite strand, things become more complex. DNA structure is antiparallel, so if one strand of DNA molecule is from 3' to 5', the opposite one will be 5' to 3'. But the enzyme DNA polymerase III only reads from one direction, which is from 3' to 5'. So a discontinuous transcript occurs. The segments created are called the Okazaki fragment, and the strand is named the ligand strand. At first, the DNA polymerase III will replicate a short episode, then it leaves. RNA primer will put a short episode of RNA onto the template DNA as the starting of transcription. Then the DNA polymerase I replaces the RNA primer with DNA segment by transforming RNA long range into DNA long range. There will have a little gap between them, but an enzyme can cope with it, which called ligase. It fills the gaps between Okazaki fragments, and the new strand was formed. Well, we could actually artificially synthesize DNA in labs. PCR is a technology used to elongate the template DNA strands and made a million copies of it. The most valuable application of PCR in medical laboratory is the diagnosis of infectious disease. Chromosome is composed by protein and nucleic acid. How can we define the DNA as the hereditary material, but not the protein? Hershey and Chase used bacteria and virus to determine it. Because DNA contains phosphorus but not sulfur, which protein contains. Through this way, we can verify DNA is the hereditary material. We call it Hershey-Chase experiment.
Now let's take a look at transcription. Well, transcription is the process that a sequence of DNA, which we call gene, is copied and transformed into a complementary RNA sequence by RNA polymerase. Since the transcription process occurs in eukaryote and prokaryote cell are quite different, we're going to discuss them separately later. For now, we're just going to look at the component of transcription. First, let's introduce the concept of promoter. It is a sequence of DNA that combines with RNA polymerase and starts the process of transcription. Unlike DNA polymerase, RNA polymerase doesn't need any primer to start to transcribe. Usually, it is the transcription initiation site within the promoter and terminator, which is a sequence that ends transcription, besides the process of transcription. The whole process can be divided into three sections, which are initiation, elongation, and termination. Most prokaryote cell has something called the operator. It is within the promoter sequence which controls the binding of RNA polymerase to promoter and forms the transcription initiation complex, which we call TNC. The operator promoter together with the coding region that was being transcribed are called the opron. So, how do they actually work? Initiation section. Once the switch of operator was opened, RNA polymerase will bind to the promoter, causing DNA to unwind and separate into two strands. Then enters the elongation section, which the RNA polymerase starts to transcribe the coding region within the opron by complementary pairing three nucleotides, so-called DNTPs, to their opposite partner on DNA and form a new RNA strand in a 5' to 3' direction, which A is paired to T and C is paired to G. However, it is no T in RNA sequence, so it is replaced with uracil, which is new. Of course, the switch of the operator can be closed by repressor, but we are not going to talk about that. Termination occurs after the RNA polymerase reaches the terminator sequence. It, together with the RNA strands formed, will detach from the DNA strand, and the DNA molecule will reannul. The strand that's transcribed into RNA sequence is called the antisense strand, and the one who isn't transcribed into RNA sequence is called the sense strand of DNA. Well, then in eukaryote cell there's something different. First,
We don't have operator. We don't have operator, and we don't have operator.
sad, isn't it? But fortunately, we got something else called the transcription factor, which will bind to the regulatory sequence on DNA that's next to the promoter. We know that once the RNA polymerase binds to the promoter, will form a TIC. Then the RNA polymerase starts to transcribe and form a new strand of RNA and wait to detach until it reaches the terminator. However, the terminator in eukaryote cells are actually a sequence, AUAAA, which will lead to the polyadenylation of mRNA. But that's not the end of gene transcription. There's something called the post-transcriptional modification, which occur both in eukaryote and prokaryote cells. But here we are going to only talk about the modification of RNA, which only occur in eukaryote cells. Well, there are three events that must occur to make a mature mRNA. First, a methylate cap was added to the 5' end of the RNA strand, which provides protection against degradation by exonuclease and allows the strand to be recognized by cells' translational machinery. Then, a long chain of adenosine nucleotides, so-called the poly A tail, was added to the 3' end of the RNA strand, which improved the stability of RNA transcribed, and this is called polyadenylation. Thus, there are many non-coding sequences within the gene of eukaryote cell called intron, and what is finally expressed is actually the extron. These introns were cut off from the sequence by spliceosome, and extrons fused together. Now we have an entire matured mRNA that's ready for translation.
So the main idea of translation is to convert RNA into protein. The translation process involves several phases: initiation phase, elongation phase, translocation phase, and termination phase. Once mRNA is produced from the DNA template, the process of actually producing the protein at the ribosomes can begin. So first, I'm going to talk about something about ribosome. Each ribosome consists of a large subunit and a small subunit. The subunits are composed of ribosome RNA, which is RNA molecules, and many distinct proteins. Prokaryote ribosomes are smaller than eukaryote ribosomes. There is also a difference in molecular makeup. The decoding of the strand of mRNA to produce a polypeptide chain occurs in the space between the two subunits. In this area, there is a binding site for mRNA and three sites for the binding of tRNA. Each that sets from which tRNA that has lost its amino acid is discharged. P set hosts the tRNA carrying the growing polypeptide chain, and A set hosts the tRNA carrying the next amino acid to be added to the polypeptide chain. And also, there are 46 possible codons. 3 codons have no complementary tRNA anticodons. They are the stop codons. There is a start codon AUG that signals the beginning of the polypeptide chain. Initiation phase. To begin the process of translation, an mRNA molecule binds to the small ribosome subunit at an mRNA binding site. Initiate a tRNA molecule carrying the start codon AUG. Then the large ribosome subunit binds to the small one. The initiator tRNA is in the P site. The next codon signals another tRNA to bind. It occupies the A site. A peptide bond is formed between the amino acid in the P and A site. Elongation phase. This phase involves tRNAs bringing amino acid to the mRNA ribosome complex in the order specified by the codons of mRNA. Ribosomes called the elongation factor assign in binding of tRNAs to exposed mRNA codons at the A site. The initiator tRNA moves to the P site. The ribosome catalyzes the formation of peptide bond. Termination phase. The termination phase begins when one of the three stop codon appears at the open A site. A protein called the release factor then fills the A site. The release factor does not carry amino acid. It catalyzes hydrolysis of the bond linking the tRNA in the P site with the polypeptide chain. This frees the polypeptide, releasing it from the ribosome. The ribosome then separates from the mRNA and splits into its two subunits.